Fabulous. Well, let me just start by thanking the organizers for inviting me. It's really an honor to be at this beautiful venue in this beautiful city and to be surrounded by so many outstanding scientists and to hear all these amazing talks. It's really a fascinating collection of topics and themes that we've been already exposed to, and I'm looking forward to the remainder of the meeting. So I am a computational neuroscientist at Princeton University, and I'm going to tell you about some recent work from my lab in collaboration with Aditi Jha, who's a former PhD student who's now a postdoc at Stanford with Scott Linderman. All right. So I'd like to tell you about this one story. All right, so the high-level motivation. So I should say I'll start by motivating the problem we're going to try to study, and then I'll dive into the details of how we tried to attack it. But so the problem we often face, problem we often face in computational neuroscience, systems neuroscience, is we collaborate with someone who's able to record from a large number of channels in the brain, and so we get this high-dimensional recording of raw neural activity. It might be spike trains, fMRI, calcium imaging, etc. And we want to make sense of that activity to understand what's being computed in different parts of the brain. How is information flowing between brain regions, or what dynamics exist in each brain region? And so a popular tool for attacking that problem is to try to fit some kind of latent state model. All right, so we saw Juan Gallegos gave a very nice treatment of manifold models that underlie low-dimensional manifolds that underlie high-dimensional neural activity yesterday, and we heard a very nice talk from Ayesha Vermani from Meming's group, who talked about a new comprehensive way to do that, to stitch together recordings from multiple brains and multiple sessions. But the common theme remains, which is that our goal is to explain this high-D neural activity in terms of some low-dimensional dynamics that we have a hope of understanding. All right, so we're going to effectively fit these models consist of two different parts. At the top, there's a dynamics equation. So this low-dimensional variable we'll call x, and we think that x at time t minus 1 progresses to x at the next time step via some nonlinear function f, perhaps, the dynamics function. And then there's a mapping from this low-dimensional latent space, sorry, I'm still learning to use the pointer here, from this low-dimensional space to the high-dimensional neural activity, and that happens via an observation equation. So the mapping from x at time t to y at time t, the high-dimensional vectors of neural activity, happen by some other equation, all right? And so this is a very popular approach to studying the brain, going all the way back to linear dynamical systems, in fact, will be the main focus of my talk today. But I want to highlight some of the limitations of this kind of approach, which is that it's generally approached as a statistical problem. We want to just predict these patterns of high-dimensional activity, or be able to kind of understand their dynamics. But that leaves us with an important problem, which is we don't know how to map that kind of statistical level description onto the underlying mechanism. So I would argue that many times these dynamics equations, right, how the activity progresses in time is formulated in this abstract manifold, which doesn't tell you how you would go building that out of neurons, all right? So we get limited insight into mechanism. And the second problem is that these models often have an inability to account for the kinds of perturbations that we're able to do with modern neuroscience tools. So in particular, we're now able to perturb specific populations. So you might have an optogenetic perturbation experiment where you're able to, let's see. Yeah, at some time you're able to turn on a laser and silence some subset of the neurons. So let's say we turn on the laser at time t here, and we silence all these excitatory neurons, and that has some effect on the other neurons in the same population. Let's say we have separate populations of excitatory and inhibitory neurons, and that perturbation pushes the activity maybe into some new region of state space that we hadn't observed previously. But more importantly, this kind of latent dynamical system model that we fit may not be able to tell us what's going on here. In fact, often it's very hard to make sense of these kinds of perturbation experiments because this may be a region of neural activity space that we haven't seen before. And moreover, the direction of causality in these models is the wrong way around, right? This model tries to model the dynamics in this space and then project that up to the high-dimensional space of neural activity, whereas these perturbations are actually happening in neural activity space. And so mapping them back to the latent dynamics may not be obvious how to do that. Okay, so the high-level theme is, how do we take these statistical models that are so powerful for giving us insight into how the brain works, how do we map them onto experiments where we happen to have cell type information? So can we take these models and make them one step more biologically plausible by incorporating cell type information, and we hope that doing so will allow us to then explain things like cell type specific perturbations. Okay, so the general question that motivates the talk is, can we construct latent dynamical systems of the kind I showed you on the previous slide that take into account cell types? Okay, so let me start by giving you a little bit of background. So the background, we'll start with the very simplest kind of latent dynamical model that has been used to describe neural data, namely linear Gaussian dynamical systems, all right? So as I said before, these models are defined by two equations. We have a low-dimensional latent variable x that advances through linear dynamics. So the activity at time t+1 represents the activity at the previous time step times the dynamics matrix A. So A describes how the dynamics unfold in time. And then we have an observation equation, again linear, that tells us how these latent states project up to give us patterns of neural activity. All right. Okay. And if you want to think about what this model looks like, right? So what are these parameters, A and C? And I should say we also assume that there's additional noise in these models that account for model mismatch and the fact that the dynamics may not be deterministic from moment to moment. All right, so you can think of the columns of this C matrix as the principal components of the neural activity, right? Any arbitrary pattern of neural activity has to be made up as a linear superposition of the columns of C. So if I just did PCA on my activity, I would expect those principal components to look like the columns here, all right? But there's and then moreover, the dynamics matrix here would look like how those principal components trade off over time. Maybe one principal component, high perturbation of one principal component trades off and projects onto another principal component, or maybe they decay away in time, or we could have a system that is exponentially growing over time. These A matrix is gonna try to explain the evolution of this neural activity in terms of those low D dynamics of the principal components. Okay, so that's the intuition to have about how these models work. But there's a major problem, which is that if I just take data recorded in the brain from two different cell types, so for most of the talk, I'll focus on, assuming we only have two kinds of cells, an excitatory population and an inhibitory population, but the approach I'll describe is general. You can apply it to other settings where we have arbitrarily many different kinds of cell types. But typically, if I show you what those principal components look like, they consist of loading weights onto vectors of population activity, and often they'll be loading onto both excitatory and inhibitory neurons equally, and with opposite signs. So in other words, this particular principal component has, if I turn it on, it downregulates these I neurons and upregulates these I neurons here. It doesn't affect only one population, all right? So it's very rare for the principal components to be able to explain just a perturbation to only the E cells. Okay, so that's one of the problems that we want to overcome, is to be able to understand how a perturbation to one cell type could be modeled with this kind of a system that I've drawn out on the right here. Okay, so our solution to that is something we call cell type dynamical systems, and it consists in really taking that linear dynamical model that I told you about and restricting the parameters to make them interpretable in terms of underlying cell types, okay? And so that consists of two basic steps. The first is that we're gonna assume that we have distinct latent variables for each cell class, okay? So you can see those here. I'm gonna have some I latents here in blue, which just affect the activity of these inhibitory neurons, and I'll have some excitatory or E latents here, which only map to these E responses here, all right? So in order to give the model that kind of structure, what we need to do is we need to constrain this C matrix here, the projection from the latent variables up to the vectors of high-dimensional neural activity, to have zeros on the off-diagonal here, all right? So that means that I have inhibitory latents here, the blue 3 dots here, only affect these inhibitory neurons, and similarly the excitatory latents via this block here will affect the excitatory neurons, and the zero off-diagonal blocks mean that there's no crosstalk between latents and cell types that we recorded. And the second thing that I'm going to require is that these blocks are positive, because I want the effect of a perturbation of the population to all have the same sign. So in other words, if this inhibitory latent variable goes up, I want that to mean that all of the inhibitory neurons that load onto that latent variable, all their firing rates go up. So in other words, when I step on, I suppress the activity of a bunch of excitatory neurons here. I want to have the same effect. I want to be able to interpret that in terms of a single latent variable being pushed down, all right? I don't want one latent variable going up to have some neurons being up and some neurons going down, okay? So we have these sign and block sparse constraints on the C matrix. And the second feature of the model will be a constraint on the dynamics matrix to enforce Dale's law. Okay, so what does that mean? As I told you, we have our latent variables divided into excitatory and inhibitory latent variables, and if we want to make those distinctions meaningful, then what we need to have happen is that the inhibitory latent variables should have a negative sign in the A matrix here, so that they can only give an inhibitory effect at the next time step. And The right half of this matrix should have only positive weights because those refer to the weighting on the excitatory latents. So in other words, the excitatory latents should only be able to excite all of the latents. The inhibitory at the next time step. The inhibitory latent variables should only be able to inhibit or suppress the latent variables, both E and I, at the next time step. Okay? So with those two constraints in mind, we now have a new model family called CTDS, which we can map more directly onto recordings with different cell types. And I'll point out one way in which we can extend the model further. We can make this a model for multiple regions, right? So imagine now that I have a recording from 2 different brain regions. Both of those brain regions could have E and I latents in them, which projects specifically, so the latents for region 1 would project only to the region 1 data, et cetera. But we might, moreover, know something about the way that these 2 regions talk to each other. So in particular, we might want to constrain the dynamics matrix. Often it's the case that cross-region projections are carried only by pyramidal cells, by axons from excitatory neurons. So we might want to say that these blocks here, which would project from the inhibitory population in region 1 to the neurons in population 2, these weights all have to be 0 here, which would refer to the cross-region weights from the inhibitory latents. All right, so we'll only allow excitatory neurons to project across regions. All right, so that's an additional, if we know something about the connectivity arising from the connectome, we can put that into our model in various ways through structure in the C matrix and the A matrix, as I've shown you here. I just want to highlight a connection to one very closely related piece of work from Guillaume Hennequin's lab. They have a very nice bioRxiv paper from 2024, which does very much the same thing as us. I was dismayed when I saw this paper because in many ways it does a nicer version of what we did. They actually have nonlinearities in their model, but they have many of the similar features, E and I cell types, low-dimensional latents that try to explain these high-dimensional data, and they're able to also account for perturbation. So there's some differences in the way they approach the problem that lead to slightly different strengths and weaknesses, but I encourage you to check that out. It's an alternative approach to ours that I think is also very, very useful and very powerful. Okay, so let me now talk about the problem of fitting our model to data. So in general, we'll start with some data that we'll assume come with some labels. So hopefully you're working with someone who's tagged some of the E and I cells, or in the data that I'll tell you about, we'll mostly be relying on spike width to know that narrow spikes happen to be putative inhibitory neurons, and cells with wider spikes tend to be putative excitatory neurons. It turns out that fitting the model, so we want to infer these parameters, the dynamics matrix A and the coupling matrix, the observation matrix C, as well as the noise covariance Q and R affecting the observation noise in this population and the latent dynamics noise. These are the parameters, and it turns out one virtue of having this model still be within the family of latent linear dynamical systems is that we can do exact fitting via expectation maximization algorithm, where it turns out that the M step just involves adding these constraints on positivity and zeros, and so you can solve the M step with a quadratic programming problem instead of the closed form updates that you normally get for EM. If that means something to you, then that's a nice feature to have. If it doesn't, that's okay. It's not important. All right. One other aside that I'll mention, just in case you're curious about the relationship between different parts of the field, we actually realized that there's also an exact mapping to low-rank EI recurrent neural networks. So there's been a lot of interest in the recent literature, in particular from Sergen Ostoyic's lab, in fitting models of the following form, where we have direct low-rank connectivity from a set of neurons to the neurons at the next time step. So no latent variables at all, but this J matrix happens to be low rank. And if we additionally add a Dale's law constraint on the J matrix, and we assume that the system is noiseless, then we have a one-to-one mapping onto the, it turns out, to the cell type dynamical systems model that I just described to you. And it turns out, interestingly, that the fitting problem here corresponds exactly to non-negative matrix factorization. So we can go back and forth. I can start with this kind of model and flesh it out as a recurrent neural network model, which looks more like a mechanistic model, because we have the J matrix would be the pairwise synapses between every pair of neurons. But if we want to interpret the model, it's easier to do that in terms of these low-D latent variables that we get from CTDS. Okay, so that's enough of the theory. That's the model that I want to propose to you. The next step is I want to show you some applications to neural data. In particular, we're going to apply it to 3 different perturbation experiments that were performed over many years in the Brody lab, and I want to show you sort of what is the model bias in terms of being able to explain the effects of neural perturbations. So let me start by describing the data set that we will be examining. Behaviorally, these were rats that were trained to perform a task called the Poisson-Clicks task. This is a now famous task in the field, which I think was started by Bing Brunton when she was a PhD student in Carlos Brody's lab. Very briefly, if you haven't seen this before, the rat pokes its nose into a port, and it's presented with a series of clicks on the left and the right. So there'll be clicks on the left side, clicks on the right side, and the animal's task is to decide which side has more clicks. So if there are more clicks on the right, the animal has to then poke to the right-hand side to get a water reward. If there are more clicks on the left, the animal has to poke to the left. All right? So that's the basic structure of the task. And the data set that we will be using to analyze here comes from some dual region recordings that were carried out by Diksha Gupta, who is a former PhD student in Carlos's lab. She's now a postdoc at the Sainsbury Wellcome Center. And what she did is she made neural pixel probe recordings in 2 different regions in the brain. So she has this neural pixels probe that penetrates through cortex, and she recorded a group of neurons in the frontal orienting fields, or FOF. That's cortical, and then it descended into the striatum, a part known as the anterodorsal striatum, or ADS. All right, so she has these 2 populations recorded, and what we're going to do is we're going to explore whether the model that we fit to these recordings can account for 3 different kinds of perturbations, which were actually performed in 3 different papers that have been published by the Brody lab. So the behavioral data that we'll be looking at come from a series of different papers of the last 10 years. So in particular, they have in the past performed inactivations of the excitatory neurons in FOF, inactivations of the striatum has only inhibitory neurons in it, so inactivating those inhibitory neurons in the striatum. And then more recently, the novel contribution from Diksha is that she was able to inactivate the axon terminals here in the ADS. So you're not perturbing either population directly, you're only turning off the axons that would be incoming from the cortex into the striatum. Okay, so that's our goal, is to see if we can account for those 3 different kinds of perturbations, how they affect the animal's behavior, using only neural data that we use to fit the CTDS model. Okay. All right, so our approach, I'm going to describe the logic of the approach, and then I'll show you the results in a series of slides. So the first thing that we're going to do is we're going to fit the cell type dynamical systems model to unperturbed data. And I should say this is partly a consequence of the way the data were collected. So Diksha made the neurophysiological recordings with no perturbations, right? So she records neural activity in both FOF and ADS, and you can see an example correct left trial from, let's see, 85 neurons in FOF and 67 neurons in the striatum. This is a correct left trial. Here's a correct right trial. I would submit that if you just stare at these by eye, it's not that easy to see which is which, right? It's not obvious that there's a larger spike rate on one side or the other. If you look in detail, it turns out that on average, the neurons fire a little bit more for right trials than left trials in this experiment, but you'd have to squint very carefully to look at that. Anyway, so she made the recordings of these 2 brain regions in animals that were performing the task, but there was no optogenetic inactivation. She later then was able to optogenetically perturb the axon terminals in the striatum and measure the animal's behavior, but not to record neural activity, all right? So the way we'll analyze the data is we'll start by fitting the model to just the neural data that that Diksha recorded. Then we're going to train a decoder. Ultimately, we want to be able to link the latent variables that we extract from our model to the animal's choice. And to do that, we will train a logistic regression classifier, which will allow us to map those latent variables onto choice. So we'll have a set of weights that will pass through a sigmoid nonlinearity to give us a left or right decision on the basis of that low-dimensional latent variable. We'll fit that model using the animal's choice data once we've already fit the latents from the CTDS model. And then lastly, in a novel data set, Diksha was able to perturb these axon terminals, but we'll look at perturbations in FOF and ADS as well. Can we now predict what happens when you perturb one of these populations and not the other at various times? Okay, so that's the logic of what I'll show you next. First, let me show you how we fit the model to data. So of course, there's a question of how many dimensions do you need in your latent variables? How many dimensions do you need to capture the neural data quite well? Here is a plot of the performance capturing single neuron responses as a function of the number of latent variables per population. So we have E and I neurons in the FOF, and we have only I neurons in the striatum. And you can see we get up close to around the train test correlation once you get to around 5 or 6 latent dimensions. However, the decoding accuracy, our ability to predict whether the animal would choose left or right on each trial, doesn't continue to increase really even beyond 1 or 2 dimensions. So in practice, what we're going to do to make things simple, we are going to focus on the 2-dimensional model just because that allows us to make 2-dimensional plots of the latent dynamics. We'll focus on 2 dimensions, which aren't quite as good as using a higher dimensional model, but it'll allow us to tell a simpler story about what's going on in the latent dynamics. And we can see that we're not really losing anything in our ability to capture the animal's behavior. Okay. So I'll focus on the two-dimensional model, and let me show you the parameters that we extracted when we fit this model to the data from FOF and ADS, okay? So this is a portrait of the dynamics matrix that we fit for one particular recording, and you can see here this upper block is the excitatory and inhibitory weights for latent variables within the cortex, all right? So this upper block would be E to E weights. These are I to I weights. Just within FOF would be that upper block there. This lower block here would be the connections within the striatum. And then we made the assumption these positive weights here would be the connections from FOF to the striatum. They're only excitatory neurons that would make that monosynaptic projection. And there are no inhibitory neurons making feed-forward projections to the striatum, so we set all those weights to 0. Conversely, the projection from striatum back to cortex is polysynaptic. So typically, we'll go through multiple brain regions before getting back to cortex. And as a result, we didn't constrain these weights in the opposite direction because we wanted to allow the striatum to have both excitatory and inhibitory influences on the cortex, all right? So that's what the dynamics matrix looks like. If I had more time, I would show you more about the portrait of the dynamics. It turns out that there's one eigenvalue of this matrix, which is slightly above 1, which means that the dynamics of the system are slightly unstable. This corresponds to the fact that neural activity over time tends to rise. Even in the absence of input, the average firing rates as a function of time tend to rise gradually. And it turns out if we decouple the striatum from the FOF, the eigenvalues fall below 1, and we get a stable dynamical system. So mildly unstable dynamics. Let me show you a portrait of the dynamics on a single trial with strong leftward evidence and strong rightward evidence. And so staring at this, it's not that obvious what has changed, all right? We can see that the excitatory latents here in the FOF diverge maybe from each other more strongly in a strong rightward evidence trial than a leftward evidence trial. However, when we look at the classifier weights that would allow us to predict the animal's behavior from these latent variables, we see that the weights are actually summing together these two latent variables. So the sum of these two latents will be what drives our choice about left versus right. And similarly, the striatal readout weights are also both positive here. So we'll be averaging these two latent variables to get the animal's prediction of the animal's choice. If we project these time-varying trajectories onto these classifier weights, we get a one-dimensional decision variable, which will allow us to predict the animal's choice for a single trial. So for this particular example trial, the latent variable stays on this side of the left choice axis, whereas on the strong rightward choice, these inputs drive the latent variable across the separatrix here to predict a rightward choice. Okay, so this 1D portrait allows us to now manipulate different aspects of the model and to see what would happen on the animal's predicted choice. Okay, let me now quickly show you how that looks. So it turns out that in a series of experiments from the Brody lab, they have, oh, sorry, I need to go back one slide. They have practiced inactivating either during the first half of the stimulus period. So this is when the clicks are on, right? You have clicks on the left and the right, and we might want to interfere with the first half of the evidence that's being accumulated, or interfere with the second half and see whether that has different effects on behavior. All right? So the first paper from this series came from Tim Hanks in 2015. And let me show you a prediction of the model now when applied to that kind of perturbation. So here, Tim perturbed, he silenced the excitatory neurons in the FOF, both early and late, and we want to see what that looks like. So first of all, if we silence during the first half of the trial, this is a left evidence trial again and a right evidence trial, we see that the choice variable still ends up on the right side of the separatrix here. So it ends up that there's very minimal effect on choice. On the other hand, if we silence late in the trial, the latent variable here is perturbed much more positively. So this latent here is very close to the separatrix, and on right trials, the animal's much more biased to make left choice errors, all right? So late perturbations, but not early perturbations, can cause large errors in the animal's behavior. So on the right-hand side, you see the data from the Hanks paper. Early perturbations have a very small effect on the animal's choices. They're biased, and late perturbations have a larger effect. Here's the predictions of the cell-type dynamical systems model that we fit, again, remember, using only the unperturbed data. We see that when there's no perturbation versus an early perturbation, we have relatively small changes in error rate, but large changes in error rate when the perturbation is made late, matching the pattern observed in the neural data. Just to show you, if I had instead tried to do this with the standard LDS model. So it's not obvious, as I said, how to apply the perturbations just to one population using the standard LDS. And we did that by randomly selecting groups of a subset of the latent variables, but we're not able to capture this effect when we don't have the cell type information. Okay. Let me quickly show you two more experiments. The second experiment was from Michael Yartsev in 2018. He silenced the inhibitory neurons. Now he got a large bias, whether he silenced early or late. We see that same pattern recapitulated in the cell-type dynamical systems model. And then finally, in Diksha's experiments, as I told you, she was able to inactivate the axon terminals connecting the FOF to the ADS. And so one question you might be asking is, how do you flesh that out in terms of our model? Well, because we have a model which has interpretable connections in it, we can just zero out the weights in this block here, which correspond to the connections from the E latents to the coming into the ADS here. And what we found was similar to the data she observed, early and late perturbations both had a large effect on behavior. Similarly in our model, no effect when there's no perturbation, but large effects for both early and late perturbations. All right, so we find this very surprising that we were able to do this, right? Remember, the model was fit without access to any of these kinds of perturbations or to the behavior. The model was fit only using just the unperturbed neural data, and yet we're able to make these perturbations qualitatively about when different perturbations to which populations will have different effects on behavior. Final point before I stop, I just want to mention one bonus application that we can also use the model if we have a population where we don't know what all the cell types are. So you might imagine maybe you know some of the excitatory and inhibitory neurons, but you have some neurons where you don't know whether they're E or I. And so in a test experiment, we asked whether, when we withhold a fraction of the neurons, can we correctly identify their labels? And that's something that we can do well above chance. We're not at 100%, but somewhere around 80 to 90%, decreasing as we increase the fraction of held out cells. One major caveat is that this is when the true cell type classifications were based on spike width, so there's some chance that we could be wrong about some of the cell identities there. But it's something we're excited about pursuing further in other data sets. So with that, I'd just like to wrap up. So I told you about the cell type dynamical systems model, which allows us to impose Dale's law with distinct latents for each cell type and to incorporate known multi-region connectivity patterns. It captures perturbation effects in both cortex and striatum, even though it was fit to only unperturbed data, and allows us to identify unknown cell types from activity, in simulations at least. That's something we're still looking at more aggressively with actually labeled data. The future challenge, and I think this is the thing we all want to think about, we have talks, very beautiful talk from Bing about using connectomics to inform dynamics models. I know Srini Turaga is also going to talk about that. I think the high-level thing that we could extract from a lot of these talks is what forms of biological structure can we take from the brain and use them to enrich our models to make more precise conclusions about how the brain actually works. So that's, I'll stop there. Thank you so much for your attention. I just want to thank Aditi, who has led this project from start to finish. She's preparing a journal version of the paper to describe this that we're hoping to release in the next month, and Diksha Gupta and Carlos, who are our collaborators on this work. Thank you very much.
Thank you. Great talk. We have time for a couple of questions. I see Mate at the back. And then John.
Mate. He's loud. He could just yell.
I could, but I won't.